one of the things that William Osler did was bring in more bedside teaching. And the whole concept that you learn from studying patients, whether that be medical students or us as physicians. And I think, as Neil has indicated, I've spent all of my life working in Newcastle. And I've learned a lot from my patients. The reasons I got into studying mitochondrial disease was, as an SHO in neurology, I saw a patient that I couldn't really diagnose. And I think that it's very interesting how much and how medical training and medical education benefit from this, but also medical research, and I hope I'll be able to show you that. I also want to stress that there is something about being a physician that I absolutely love. I still love, having done it now for the best part of 42 years. I still love the fact that you see patients and you're constantly learning. But I also like the concept, and Mandy Burnham kind of touched on this, that I like the idea that I look after patients for a long time. I like the idea that I'm caring for them and caring often for their families. And this is just a good example. We know a lot about mitochondrial disease in Newcastle, at least in part because this young man here was somebody I studied in my PhD in 1983, and I still look after him now. So you learn a lot about patients if you look after them for that length of time. So why have I called this the small circle of life? Well, this is just a video of an event that probably happened about 2 billion years ago. The Earth's atmosphere was getting more oxygen, and what happened is a larger bacteria engulfed a smaller bacteria that had the ability to use oxygen. And it's important to recognize that that hypothesis carries on even now, because when that happened, and that engulfment happened, it also engulfed the genomic structure, what's going to call mitochondrial DNA. And that genomic structure of mitochondrial DNA resembles more like bacteria than it does our nuclear DNA. So that was 2 billion years ago. Let's bring us slightly more up to time. So it was in about the early 1950s when electron microscopy came in that we first recognized there was such a thing as mitochondrial DNA. I'm not sure how well this projects on here, but in cells it was noted that within mitochondria there was this kind of circular structure. Nobody really knew what it was. At the time they suspected it was DNA because of the way in which it came up on EM. But it was this gentleman that really showed us, this great scientist is probably a better way. There's only 4 people in science who've ever got 2 Nobel Prizes, and this is one of them. This is Fred Sanger, who got the Nobel Prize for the structure of insulin, then discovering or showing us how to do DNA sequencing. In 1981, he sequenced the first human genome, which was mitochondrial DNA. So this mitochondrial genome is remarkably simple. 16,500 bases compared to the 3 billion in our nuclear DNA, virtually no introns, virtually no junk DNA. So 16,500 bases, so what's so difficult about that? I'll show you a little bit more later. But this is a schematic representation of of the way it is in the cell. Basically, mitochondrial DNA is present in mitochondria. It's present in multiple copies, something I'll come back to later. And all it does is produce 13 proteins of the mitochondrial respiratory chain. That's all it does. But without the ability of us to metabolize oxygen, we wouldn't be here as eukaryotic organisms. So it still plays an absolutely essential role. The children we see that die within the first 48 hours of life with severe mitochondrial defects indicates just how fundamental those mitochondria are. But this is why I'm here, this is why hopefully you're listening, is because these are all patients with mitochondrial DNA disease. So these are what we've learnt since the early 1980s. These are the patients that we've learnt about that carry these mutations. And I think there are things that are very apparent there straight away. And this is why it is very interesting from a general physician's perspective. You know, patients with mitochondrial DNA disease, the youngest patient I've looked after died at 48 hours, and the oldest is well into his 80s. So it's a disease that scans the whole of the life course. The other thing that's interesting about mitochondrial disease, which from a physician's perspective, is it can involve any tissue. You have mitochondria in every cell in your body, and mitochondrial DNA. And whilst patients might present to a neurologist because the brain and the muscles tend to use an awful lot of oxygen and require a high energy input, it can affect any tissue, and literally any tissues. And to be honest, I spend more time in my clinic talking about the non-neurological problems than I do the neurological problems. So where have the successes been? I've told you that we've worked in this for a very long time. And don't get me wrong, there's other places around the world. There have been many successes in mitochondrial DNA disease. The first is, we're currently in the genomics age. Most of you will know the 100,000 genome, the whole genome sequencing. It's going to make massive differences to the way we practice medicine. There's 16,500 bases, so we've known about mitochondrial DNA mutations since the 1980s. So determining that there are mutations there has been relatively straightforward, and it continues to be relatively straightforward. We still find new mutations, and there's over 250 present in the mitochondrial genome. What is more challenging is, of course, that with nuclear genetics, we all understand that. With mitochondrial genetics, it's a different concept. You have multiple copies of mitochondrial DNA in your cells, maybe 10,000, say, in a single muscle fiber. So the concept is that all our mitochondrial DNAs are all the same and they're all normal. But many patients with mitochondrial disease have this situation where they actually have a mixture of normal and abnormal mitochondrial DNAs, and that's called heteroplasmy. And some patients actually have all mutated mitochondrial DNA. Now, why is that important? And I think it's one of the things that we've learned, and it's very important when Neil was talking about the work we've been doing about reproductive options, is because we know that the higher percentage of abnormal mitochondrial DNA you have, the more likely you are to develop symptoms. Now, it might seem obvious, but again, we've learned this specifically because we've looked after patients. And this is a family that one of my colleagues went to visit in South Wales. And I'm just picking out 3 family members. They all carry the same mitochondrial DNA mutation. So they all carry it. It's a well-characterized pathogenic mutation. And if you look at these 3 cousins, you see that one of the cousins clinically unaffected. One is mildly affected. I say mildly. She's got ataxia and some mild epilepsy. And one, as you can see, can't get up the ground because she's so weak and affected by the disease. And how do we explain the fact those 3 cousins have got the same mutation with different phenotypes? And it's fairly straightforward in the sense that the cousin that's clinically relatively mild, clinically unaffected has low level. Those with more severe disease have higher levels. It's not quite as simple as this, but this is a major factor in determining the phenotype. So what other successes have we had? Well, we've had successes. There's something, I'm not unusual in the northeast of England. We realise how lucky we are to live there, and not many of us move. So it's very good for doing population studies. And we know that mitochondrial DNA disease affects about 1 in 5,000. So that's something that's come out of work from Newcastle. The other thing which is incredibly important about mitochondrial disease for our patients is that we are nationally commissioned. And one of the slight worries I had with some of the things that Andy Burnham was saying is that, as soon as we start fragmenting up the health service, it no longer is national. And one of the great things about our service, which is a highly commissioned service, is the fact that patients from all over the country can come and see us. And that's the heat map of the patients that come to Newcastle. So there's a centre at UCL and one at Oxford, but we see patients from all over the UK. We see about 900 patients a year. So you can see that it is a highly successful system because patients have the opportunity to discuss what is a relatively rare disease with people who've actually seen quite a lot of cases. So what about other successes? Well, this is a gentleman who came to see me, age 33, presented with a seizure. And he developed a number of clinical features associated with his mitochondrial disease, exactly as I've told you. He developed diabetes, deafness, cardiomyopathy, and developed a bowel disturbance. More about that later. So one of the things about mitochondrial disease is things which seem relatively simple for other patients are less simple for patients with mitochondrial disease. Because he had a focal form of epilepsy, which you can see here, you can see on the EEG, you can see where the focus is. But because of the vulnerable brain, you can see that he had very extensive MRI changes. Now, one of the great successes that we've been able to do, and I think we have contributed a lot to this because of the unique system that we work in, is actually finding good treatments. So we've been able to write clinical guidelines for patients with mitochondrial disease about giving the best therapy. What's the best way to treat the seizures? What's the best way to treat the heart? What's the best way to treat the diabetes? What's the best way to give people anesthetics? And here he is, 23 years later. So something that could have killed him in 2 years, there he is 23 years later. So yes, we've had a lot of success. But we've also had a lot of failures. And it's the same chap I'm going to talk about in terms of the failure. And the reason why we're failing is because of people like him. And why we're failing? I've said we've sorted out his diabetes, good diabetic control. Hearing aids were fantastic. He's, everything, no epilepsy for 15 years. The reason why we failed is on your left is a normal brain scan of somebody in his 50s, and on the right is his brain scan. And you can see that despite the fact we were able to treat his acute symptoms, we couldn't stop the degeneration associated with this. Doing a bit of general medicine now, these are just his abdominal x-rays during an admission. And they are very interesting viewing because you can see that on the plain abdominal x-ray, you can see gross dilatation of his colon if you look at the image on your far right. It's just staggering. And many of these patients are admitted under the surgeons for acute obstruction because of the x-ray. So the last thing you should do is actually operate on these patients. And on the bottom images, you can see that this is just a CT scan where he's laying flat on the two left-hand images, and you can actually see the fluid levels. So in effect, what happens to these patients is that the smooth muscle in the intestine just stops working. So they get gross dysmotility. Those of you, I'm sure everybody's just looking at that CT scan on the bottom right, thinking, "What the hell is he talking about? This isn't a man." That's his bladder. So you can see that the muscle in his bladder is equally affected. So you can see just how devastating this disease can be. So yes, we've had a lot of success. Boy, have we got a long way to go. So what about the future and I'd like to say the present? So again, so much of what we do is based upon our clinical experience as clinical academics. So this is a true story that this lady came into my clinic in the 1990s, late 1990s, and that's her son who's got as you see, he's in a wheelchair. He's got a terrible degenerative disease called Leigh syndrome. But Sharon has a devastating family history. Edward is the only one of her children that survived after 48 hours of birth. So six children died in the first 48 hours, and then Edward was her only surviving son. He died about 2008 from complications associated with his Leigh syndrome. So here's this lady, and she's only one example of where this is being passed down through families. So why is it unique? Well, it's unique because mitochondrial DNA is purely maternally inherited. So we all have mitochondrial DNA, but only the mitochondrial DNA in the oocytes gets transmitted. There's no paternal contribution. So we know, for example, a man can be affected, his children won't be. A woman obviously passes it on. And that made us think back then that we've got to start doing something about this. Because I don't know, many of you will look after patients with genetic diseases, devastating genetic diseases, and the most important thing for those families is that they don't pass it on to the next generation. So can we prevent this? So what I don't want to do is I don't want to simplify this. I've obviously had to cut down on the number of slides, so I'm not going to go through all of this. I'm just going to say that giving advice to women who carry these mitochondrial DNA mutations is an extremely, extremely important part of what we do. And there are a number of different options, and I've had several arguments over the years about with doctors telling patients what to do. Your reproductive choice is one of the things that families, women, it's their reproductive choice, it's not ours. What we've got to do is present the various possibilities. So, adoption, egg donation is our options. But it's interesting because we've obviously spent a lot of time working with the patients. Most families want to have their own biological child. So there are techniques out there, such as pre-implantation genetic diagnosis, that are acceptable and possible for some women with mitochondrial DNA mutations. So I'm not saying that what I'm suggesting is suitable for everybody. What I'm saying is that we wanted to think a little bit more radically about this. And in essence, we and others came up with the concept of what is now called mitochondrial donation. And this is a fairly, like many things, it's fairly simple. So the whole idea is to take the nuclear DNA from both parents and the mitochondrial DNA from a donor woman. And the way that we do this is just after fertilization, you develop two pronuclei, which are the male and female pronuclei. You take those out of the oocyte, and then you get another donor oocyte where you've removed the nuclear DNA, and then you do the fusion. And I'm just going to show a short video of this actual technique. You can actually, with a naked eye down the microscope, see the pronuclei. And this is just one of our highly skilled embryologists just picking up one of the pronuclei that you can see there. You can see him going back for the second pronucleus, and just sucking it up. So you've now got both the pronuclei taken out. Now, when we put the pronuclei back into the donor oocyte, we've got to get those pronuclei to fuse in with the mitochondria and the rest of it, so they're washed in a kind of viral envelope protein. And you can see these just being injected back in. always just tucks them in. I'm sure she did that for dramatic effect. It's fantastic, isn't it? So you can see that this is a technique that is certainly feasible. It's very interesting because, carrying on from a kind of political side, this was illegal in the UK until 2015. This was a big move forward in actually getting this through Parliament, in both houses of Parliament, and it was, there's Jane Ellison, the previous Secretary of State for Parliamentary Under-Secretary for Public Health, signing it off. I've stressed throughout this talk of the importance of patients in what we're learning, but also, again, I just want to stress the importance of patients in everything that we do. And this would never have got through Parliament if the patients hadn't been involved. And I think that's something that we work in partnership with our patients and for our patients. So, science is great, but if you're going to try and help the patients, you've got to be moving this forward. And I think one of the challenges we've had, there have been many challenges with this. The science was difficult. Believe me, the politics and ethics were difficult. And then actually creating a clinical pathway for the patients. Because you can't just stop there. You know, we've developed this, but we have more patients waiting for it than anywhere else. So this is what we've done, is we've developed up a reproductive choice pathway for our patients. So, patients, any of your patients, anybody from in the UK can be referred into our reproductive advice clinic. And it's very much in a clinic where which is not under the same pressures that most NHS clinics, where they are giving very individual advice. We've talked about the importance of individual advice. If they want to go forward with one of the IVF options, they then go to a reproductive options clinic, where there is a mitochondrial expert and an IVF expert. Because, you might say, well, look, I want to have mitochondrial donation, but if you're too old or there's a lot of factors which will affect. You've got to be producing lots of eggs for mitochondrial donation to work. That we have to have a system for donors. This isn't going to work if we don't have women coming forward and donating their eggs. So we've got to know that those donors are healthy, that they don't have mitochondrial disease, which is hidden. We've got to look after the mothers during pregnancy. I mean, we do this anyhow because having mitochondrial DNA disease does mean that you have greater risk during pregnancy, but we need to formalize it. And most importantly, because the thing that we're doing this for is to have healthy children. So we have to have a system that actually looks at this. And again, one of the great advantages about the National Health Service is that everything on there in light green is done anyhow. What we're suggesting is that we want to do this by following up the children at 18 months. So you can have a service like this, and you can do the research, but somebody's got to fund it. And we were very fortunate that we got funding from the Wellcome Trust to fund the 18-month follow-up of the children born. So the first 75 children born will be followed up by this service. The NHS pays the excess treatment costs. And I think this is a great model by which things work, where there is combined support from both the charity and the funding. So in conclusion, I think we have a lot to learn from our patients. I think William Osler was right that the best way to learn is to actually get and see patients and to learn from our patients. I hope that we've got a lot to offer back as well. And I think that by this combined approach of both learning from the patients and doing patient-orientated research will eventually give them better care. Thank you very much.